Thanks very much. Good afternoon, everybody. My name is Baars. I work at University College Dublin. I'm a lecturer there. A lot of our research is on invasive species. My interest is in freshwater ecology as well. So I'm today going to present on invertebrates. I know this is an entomology conference, and I'm very conscious of that. But I think the threat that some of these invasive invertebrates pose is so significant that we should be aware of it in terms of conserving our other invertebrates like insects. So I'm going to be presenting a couple of case studies looking at a couple of projects that we did, looking at several aquatic, one terrestrial invasive species, and highlight the very recent introduction of the quagga mussel, which is a serious threat to our freshwater systems. So I rely on a huge group of people that are helping undergraduates, graduate students, and collaborations. So when any invasive species is threatened to come to Ireland or has arrived to Ireland, we go through a risk assessment to try and figure out what kind of level of impact or they might have. So a lot of these many of the species have been risk assessed. Quite a few species have not been risk assessed. And I would urge that many of the assessments are done based on the information that we have in terms of the ecological assessment impacts from elsewhere. So we really need more work to be done to assess the impacts that some of these invasive species have on Irish habitats and invertebrates. There's certainly a lot of work to be done on invasives. We know that they have very serious, significant threats on biodiversity. Through predation, competition, they can displace native species. And some species are ecosystem engineers, and they can significantly affect the way the system works. So when invasive species come in, there are many ways we can kind of deal with them. We can try and very often apply different techniques to try and manage these species, like mechanical control is an example of the American skunk cabbage that we've been trying to eradicate off the Liffey. But it's incredibly difficult to try and manually control something. You have to keep repeating your application. It's very expensive and labor intensive. Chemical controls, we're trying to reduce the amount of chemicals used to control invasive species. They're very effective, but there are serious ecological consequences using chemicals, and we're trying to reduce that. There are other alternatives, like biological control, where we establish natural control systems back for the species that have been introduced without their natural controls. But that hasn't systematically been applied in Europe. It works very well elsewhere, but it doesn't work for all the species. I would argue, just based on the work that we've done, that there's still a lot of work to be done to try and contain the species. Firstly, prevent species coming on. I think we all can appreciate that. But I think of the species that have come in, there's still quite a lot of work that we could do to try and prevent their further spread. And I think the Check, Clean, Dry campaign that's been run in the UK as well as in Ireland is a good example. We need to try and make sure that we don't disperse our invasive species. All too often, by the time we discover something, it's almost too late. And we've seen this in aquatic systems like the bloody red shrimp, and also the example that I'll show with quagga mussel. But they may still be confined to certain aquatic systems or terrestrial systems. So we need to be aware that, or educate people that these things can be moved to other locations and have similar impacts. So I'd like to present 3 case studies. This is Gammarus pulex. This is an aquatic amphipod that we've done some work on. It's come from UK or mainland Europe, where it's native and it's invasive or introduced in Ireland. You can see the distribution records here from the National Biodiversity Data Centre. It is quite limited to some catchments, but it is actively spreading all the time. It's a detritivore, and it also feeds on invertebrates, so it is very relevant in terms of its impact on insects. We've done some work on 2 systems draining to the east. So this is the Boyne catchment and the Liffey catchment. I just wanted to alert you to the kind of results that we were getting when we looked at these species. So here we focused on Loch Rammer, and the tributaries around Loch Ramond. It was only because we were doing some eDNA work trying to detect this new Crangonix floridanus, which was newly recorded as another invasive species in Ireland. So we're trying to find out where they were, and then we came to Loch Ramond and we found Gammarus pulex as well as Crangonix pseudogracilis. So we did some work on the lake itself, 10 sites. We did quantitative sampling. We also looked at 6 tributaries coming into the lake, and without spending too much time, these are the results that we were getting in terms of the proportion of native versus invasive. And what you can see from these pie charts, the whole lake is now completely made up of Crangonix pseudogracilis and Gammarus pulex. Gammarus pulex is going into the little rivers, but there are areas within the tributaries that are still providing a refuge for our native Gammarus dubini celticus, which is kind of their functional feeding group sort of equivalent, but they do things, I think, a little bit differently. We're not quite sure how different they are, although they've displaced the native amphipod. If we turn to the Liffey catchment, Liffey is quite strange. It rises in the Wicklow Mountains, which is on the top, sort of on the right-hand side of this graphic here, and then it goes inland, and then it works its way around to Dublin. So what you're seeing here is just what we found at 29 sites in the catchment. And Gammarus pulex was first introduced in the north and south as fish food. So they brought it into a fish farm, and then it escaped into the catchment. And you can see the white pie charts represent Gammarus pulex in the top of that catchment. Then coming into the lake, Blessington Lake is a man-made lake. It's for it produces hydroelectric. So there's a disconnect between the lake and the mid part of the river. But the lake is occupied by Crangonix pseudogracilis, totally dominant, and Gammarus dubini, which is our native, is isolated to some of the little tributaries that run into that lake. Interestingly, Gammarus pulex hasn't progressed into the rest of the Liffey. So the middle part of the Liffey and all the way down, we don't find Gammarus pulex, but we do find Crangonix pseudogracilis. What impact are these things having? It's clearly displacing some of our native species we have looked at, but what else is it doing to the rest of our invertebrates? Look at the biomass, or the abundance and biomass. These invasive species are outnumbering our natives. So where they co-occur, they're outnumbering. Where they've displaced them, they're occurring in much higher densities. So they must be doing something very different to the system. What is that impact? We're not sure. We hooked up with Inland Fisheries Ireland. They were doing a survey of the fish in the Liffey because I think fishermen were saying that the fishing stocks had decreased. So we took that opportunity to see if they were feeding on some of these invasive amphipods, and we were finding them in quite a number of the brown trout. So this is Crangonix pseudogracilis that are turning up in their diet. So we know they're being assimilated into the food web. But interestingly enough, the New Zealand mud snail was, I think, in 65% of the trout that we sampled. So that's another invasive species that has naturalized and is also being brought up into the food web. Leaving that study behind, the second study I'd like to focus on is a terrestrial amphipod. This is the New Zealand landhopper. This thing, I think, its distribution is totally underestimated. If we look at the records from the National Biodiversity Data Centre, it suggests it has a coastal distribution, but our recent records have found it in many other places, and in fact, at very high densities. This is an amphipod that lives in the leaf litter. It feeds on the leaves, and it occurs at very high density, and it's about 25% of the leaf litter decomposition is attributed to the species. So where it occurs, it certainly has an impact on the system, and then indirectly affects everything else that feeds on organic matter. We did a study in Killarney National Park, trying to assess where this thing occurs, what density it occurs, because that's the basis of how we understand their ecological impacts. We looked at 75 sites looking at the distribution of the species, and you can see these again, these little dots show, the darker dots show where they occur, and then as it gets lighter, they occur in lower abundance. But the white dots indicate where they don't occur in that survey. And you can see that there's quite a lot of sort of records around Killarney town, and then in the actual park, all around the wooded areas. They prefer the leaf litter in wooded habitats. They don't like grassland at all. So it's very difficult for them to move between sites, woodland sites. And in fact, the National Parks and Wildlife Service's rangers were saying that very often they would find fly tipping green waste in many of these wooded sites. And we reckon that this is a way that they're actually being picked up from one site and being moved to another wooded site in the area. So people think probably, what's the harm in dropping sort of green waste from your garden into a natural surrounding? But this is a consequence. So I suppose it's a good example, maybe too late for Killarney National Park, for that area, but it's a good example of how we could maybe educate people to make sure that they don't do this sort of thing that has a significant risk of spreading an invasive species. The other case study is the quagga mussel. This is something that we came across in 2021, and it's a very significant invasive species. It's one of the top worst invasive species in the world. It's a disaster that's arrived in Ireland. I think we're in a position that we maybe can try and limit its dispersal in Ireland. It looked very similar to zebra mussel, as you can see here, quagga mussel and zebra mussel. We've kind of chosen quite a light colored one. You can get them to be quite dark, so it can be quite difficult to tell them apart. You can tell them apart usually if you see a cluster of zebra mussel or quagga mussel. If you find a lighter one in that cluster, then you know that a quagga mussel is there. So in 2021, when we discovered it, we quickly ran around looking to see where it occurs, because that's, again, the basis of how we understand what interventions we can put in place. And we found it to occur in most of the Shannon system already. So by the time we discovered it, it was almost too late. But now that we've done some work last year, we now know that it's really isolated in the Shannon system. We haven't any other records outside of the Shannon system. So we're actually we have a good opportunity to not move it beyond the Shannon system. We're getting, the quagga mussel essentially creates a very dense substrate. They occur at very high densities, and we were finding densities of up to, I think, 81,000 per square meter. More typically, we'd find them between sort of 2,000 and 25,000 per square meter. So they were creating a substrate, essentially. The substrate, studies have shown, is actually quite good for biodiversity. It can increase biodiversity, but it usually replaces a habitat that isn't as complex as that. So what you're introducing is many other invertebrates that are not typical of that site where it establishes. So in profundal, the big difference between quagga mussel and zebra mussel is that the quagga mussel can establish on soft sediments. So in profundal habitats, studies have shown that quagga mussel can actually affect the species that occur there. So chironomids, oligochaetes, maybe mayflies as well. It can also displace those. So we did a survey in 2021, and what we found is that we're finding little satellite populations. So these are all the sites that we covered last year, about 240 kilometers of the Shannon system. And we pretty much know exactly where, the quagga mussel occurs. The black dots are where the satellite populations occur. And all the way further down, we see that it is well established in the Shannon system. Where it occurs with zebra mussel, it's outcompeting zebra mussel. So that it's displacing zebra mussel in the littoral habitats on hard substrates and also going onto the soft substrates. There's a set of samples here that are very significant. This is the river sites that we assessed on soft sediments where zebra mussels don't occur. And this is the kind of sort of populations we were finding. So this is a dredge sample. It's not quantitative. but it's a bucket, it's a basket that we drag. And you can see the density that we get. To do more quantitative work, we did grab samples to see what kind of densities we were getting. There, but this image is at this is at about 8 meters depth. You can see here, this is on a sort of a quadrate. And you can see the kind of densities that we're getting of quagga mussel. So you can, I mean, any aquatic ecologist will appreciate that it's definitely having an effect on the things that are surviving or trying to survive in that habitat. And it's not just a chosen, we've seen this repeatedly in many of the habitats that we surveyed. So this is the actual data from that. We were looking at seeing whether quagga mussel was establishing really at very deep habitats, and we were assessing at 5 meters, 10 meters, 15 meters, and 20 meters, 15 grabs per site. We did this at 5 sites in 3 sites in Loch Ree, 2 sites in Loch Derg. So we have a good idea of what, how it's establishing. And what we're finding is that the quagga mussel could actually form its own substrate even on soft sediment, which is quite remarkable for a species. That habitat's quite difficult, I think, for something like that to occupy. The most alarming thing is that the change that we've seen even in those 2 years, we assessed it in 2021, in 2022, you can see how all these sites are now having, so through natural dispersal, it's now moderate to high density in these sites. So that's a massive step change in a year. So we reckon, based on its the profile of the population, we reckon it was introduced in about 2016-17. So it's established to that density in such a short period of time. We did some surveys looking at the risk of spread, because if we can contain it, then at least it might have an impact on the Shannon system, but at least it won't impact the rest of our freshwater systems. In the United States, similar invasion occurred with zebra mussel and then quagga mussel, and there's every indication that the quagga mussel takes a much longer time to disperse to other aquatic systems. Whereas zebra mussels are very quick to attach something, they can resist desiccation for a lot longer than quagga mussels. So they have a greater tendency to be able to be dispersed. So we did an assessment of winterized boats just to see if quagga mussel was attaching to these things. And this is the proportion of boats that have quagga mussel on them, even winterized boats. And you can see that the proportion increased as you go further up increased to a point up to Loch Ree. And then there were a few boats that had quagga mussel on them. The most interesting ones were actually, if you look at the history of some of those boats that had quagga mussel on them, even at the top end of Shannon, they'd gone to Loch Earn for a couple of months. They traveled down to Loch Derg, and then they've traveled all the way back up to those sites where they winterized them. So the significance of that is that these things are probably in Loch Earn. We haven't yet detected it. But any unconnected water body requires them to be moved. And I think we did a survey on various different plants and see if quagga mussels were attaching to those. And we found that several plants had quagga mussels attached to them. And the significance of that is that if you've got a small boat, there's somebody fishing, pulls up their anchor, you might have this lying on the deck of your boat, and you don't, you probably won't even notice it, and you'll move to the next water body and you can dump the quagga mussel there. So there's a very high risk of these things moving. So I think, in terms of invasive species, I think it's always a bleak picture when we see another invasive species come in. But I think, we do have an opportunity to try and limit their impact. And I think this idea that once it's in the country, it's gone, I don't think that's really a good, maybe a good premise to have. I think there is, we can focus some work to try and reduce their spread to other sites. We need to inform the general public. We need a campaign of education. We need to implement the check, clean, dry campaign wherever we can, and try and promote this idea that we need to limit where these invasive species have occur. And I suppose there's also a call for more funding to try and direct more impact studies on invasive species. because I think we know very little about the actual impact they have, particularly on insects and aquatic insects in particular. Yeah, thank you very much. These are all funding.
Freshwater shrimps measurements to reduce the population of our so, do you have any thoughts on that? Are we approaching this and trying to reduce the population?
Yeah, I suppose when you try and reduce invertebrates where they are established, I think it's probably very little, there's very few mechanisms there to try and reduce the likes of amphipods and things like that. I think all that is left for them is to make sure that we're not dispersing them. So whoever's using the water amenity, that they're not relocating the invasive species to another location. Yeah.
Okay. I'd like to ask a question about trangonics.
Yep.
I've been studying beetles in pen habitats across Ireland for many years, and it's an extremely common species in dense fens with almost no visible surface water. Is it possible, you were talking about two species, Floridanus and Plecoptera. Could there be a differentiation in habitat window, or are all of these pseudograsslands?
So we've done a bit of work. We do have Floridanus in Ireland. We discovered that in 2020. We've done some work last year looking at the eDNA to try and detect Floridanus as opposed to Pseudogracilis, and it seems to be restricted to the Liffey and the Barrow system and the interconnecting canals, which brings up the question whether it's everywhere, because once it's in the canals, it's connected all the way to the Shannon system. So I'm referring it as Trangonic Pseudogracilis, but it could be Floridanus equally. Although we did take DNA samples across most of Ireland, and for those, they are mostly gracilis. And Floridanus seems to be only restricted at the minute that we know of in the Liffey and the Barrow system. Yeah. But they're very difficult to tell apart morphologically. So we'd have to, yeah.
Yeah, Ken.
Sure.
Sorry. I've also been recording the New Zealand landhopper across Ireland. I have,
okay.
I don't know, 50, 60, 70 site record. It really is incredibly widespread, but the maps don't show it.
Yeah.
So I agree with your sentiment. The maps do underrecord incidents. And regarding Clarke, I was in Clarke for a while in 2011. There was no sign of it in 2011, none whatever.
Okay.
I was speaking to the rangers about it, and they said here a couple of years ago, it spread at an astonishing rate in Clarke within the last 3 or 4 years.
Yeah, great. I wonder if we might combine the records for the species to put something together. I didn't mention, but the densities we were getting was up to 4,000 per square meter. As soon as you move the leaf litter, they just everywhere.